This means that these polynucleotides comprise of different mononucleotides. And as you see in this picture, you can see mononucleotides. Each mononucleotides comprise three sections. One section is carbon sugar or five carbon. The second section is phosphate group, and the third section is bases. Four alternative bases. In this picture, you can see the five carbon sugar. As you see, in two carbon sugar, sometimes we have not oxygen group. In carbon sugar, sometimes we have not oxygen group. And you can see a five carbon without 2' oxygen, five carbon sugar. Name is deoxyribose. That name is deoxyribose. And this carbon sugar has in the 2' carbon. That name is, and carbon sugar called as ribose. For you two different concept in carbon sugar. Deoxyribose. But what does mean deoxyribonucleic acid? Why we call it deoxyribonucleic acid? We call it nucleic because it's found in the nucleus. It's found in the nucleus. And why we call it acid? Because it has acidic chemistry characteristic. Acidic chemistry characteristic. In this picture, you can see the ribose. In this picture, you can see ribose and deoxyribose. In ribose sugar, as you see, you can see the oxygen prime carbon, and in deoxyribose, you can't see the oxygen group in the 2' carbon from carbon sugar. Therefore, that is ribose, and that name is deoxyribose. DNA and both have four bases, 2' and 2' pyrimidines in their nucleotide chains. Both molecules have the purine, adenine, and guanine, and the cytosine. DNA has a pyrimidine, thymine. RNA has the pyrimidine uracil. RNA has the uracil. Thus, three of the nitrogenous bases are found in both DNA and whereas thymine unique to DNA and unique to RNA. Did you understand this? Did you understand the structure of the nucleotide? My friends, did you understand the structure of DNA? Note everything that you want in your notebook. Okay, note everything. And please writing everything in your notebook. I want to stop each slide for 1 minute because you should writing everything in each slide in your notebook. Okay, please writing everything in your notebooks, and I'm stopping on each slide for 2 minutes. When you writing everything, please let me know to continue the lesson. Okay? Okay. Your time starting. Your time start. No. 2 minutes. You have a 2 minutes for writing everything in this slide in your notebook. لطفا بچه‌ها من برای هر اسلایدی 2 دقیقه صبر میکنم تا داخل اسلاید رو تو دفتراتون بنویسید که اگر به هر دلیلی نتونستیم فایل رو به شما برسونیم شما مطلبی برای خوندن داشته باشید. خب دقیقا چی گفتم؟ بسیار. و هر جا که سوال میتونید از من بپرسید. In it, you can see the structure of nucleotides. In this slide, you can see the structure of nucleotides. This comprised of three units. One unit name is phosphate group, another unit is sugar group, and another is nitrogen containing bases. Nitrogen containing bases. And you in this slide you can the structure of nucleotide and mechanism of different sections or different units together, or the maps of the binding of the section of the You're now unmuted. Okay, you have 2 minutes for writing everything in your notebook. Please write the structure of the acid. Please write the structure of nucleotides in your notebook. The map of different section and kind of and different kind of binding these sections. Okay. In this picture, or in this slide, you can see different bases and different map of and different structure of nucleotides, and you can see different kind of the carbon sugar with three different of nitrogenous bases, no nitrogenous bases. Some of them are pyrimidines and others are purines. What is the pyrimidine nitrogen bases? The pyrimidine nitrogen bases are uracil and thymine. And the purines are guanine and adenine. In this picture, you can see again the five sugar cup. And in this picture, you can see the figure of the structure of nucleotides. چرا این مبحثی خدمتتون رو درس دادم و انتهای کلاس براتون میذارم اما با این وجود بازم بنویسید برای خودتون بچه. I will send you the PDF of this lesson, but please writing everything in your notebook, because writing help to better understanding difference Okay. As you see in this picture, in this section, you can see the nucleoside. You can see a nucleotides. The nucleotides comprise three sections: nucleoside, nucleoside monophosphate, nucleoside diphosphate, and nucleoside triphosphate. What was the nucleoside? The nucleoside is or nucleoside of only a carbon sugar and a nitrogenous bases. Please writing this picture in your book.
You're now unmuted.
Okay, I know the sound is very bad. Okay, but I'm stopping each slide for 2 minutes. Please, everything in each slide. And besides, I you a book biotechnology, a book of the DNA about and you can read and find and therefore you can everything in this class. Okay. You write everything? As you see in this nucleotide compound of three phosphate group. Therefore, each nucleotide comprised of 3 phosphate group. The energy held in the extra phosphate is used among other purposes. Note TP, adenosine trait. The energy of this is nucleoside phosphate. Okay, please write everything in this slide. In this slide, I want to teach you about nucleic acid nomenclature. یا نامگذاری اسیدهای نوکلئیک. Nucleic acid nomenclature. What is the first column? The first column is the name of different or alternate of the bases. Adenine, guanine, cytosine, thymine, uracil, and adenosine in DNA and RNA have a different form of nomenclature. DNA that deoxyadenosine, and in RNA is that name is adenosine. In the DNA is deoxyguanosine, and in RNA is guanosine. Deoxycytidine and cytidine. Deoxythymidine and thymidine. As I mentioned you, in RNA we have not a type. But you have a uridine. In RNA you have a uridine, and in DNA you have a uridine. Okay, thymine and urine and uracil or uridine are replaced DNA and RNA together. In DNA, deoxyadenosine prime 3-phosphate DATP. In deoxyadenosine 5'3-phosphate, guanosine 3-phosphate or DGTP, or deoxycytidine 5'3-phosphate and deoxythymidine 5'3-phosphate. But adenosine 5'3-phosphate, 5'3-phosphate, and cytidine 5'3-phosphate are the nomenclatures for calling DNA in nucleic acid. Please write this your notebook. As you see in this slide, the bond joining the 5' of the deoxyribose sugar to the heterocyclic bases is the endoplastic bond. Rotation above this bond gives rise to syn and conformation. Therefore, above this bond gives rise to syn conformation. Rotation response is restricted and the anti-conformation is generally favored. Again, rotation about this bond is restricted and the conformation is generally favored. Anti, and anti, and a different type but nucleotides. You can see deoxyadenosine as a pre-nucleotide. Deoxyadenosine in anti-conformation and in sync conformation, or deoxyadenosine in anti-conformation and in sync conformation. Please write this. Write it in your papers or in your notebook. Okay. In the dot place, adopt the anti-conformation. Again, this nucleoside in the DNA dot place adopt the anti-conformation. There are very few exceptions to this rule, which is guanosine monophosphate, in which the guanine base adopt the conformation about the electric bond. The 1' position of the nucleodeoxyribose sugar, the anomeric. If a substituent attached to the carbon lie on the same face of the sugar ring as the hydroxide group, known as the beta ionomer. It is known as the beta ionomer. If a substituent is one of the opposite side of the sugar ring, it is the alpha ionomer. Alpha ionomer and beta ionomer. Nucleoside in A are the beta configuration. Beta ionomer is the most or popular conformation of the nucleotides in the DNA. Okay, please, or nucleoside in the DNA. Please write the information in this slide in your paper or notebook. Paper, my friends, please write every paper. Okay, can I continue? Did you write everything? Of work, scientists had labored on their enormous hypothesis. This has proved that DNA made of a quantity of the four bases. Therefore, subunit of this DNA contains one copy of each basis. Of ratio until charge of work and scientists had the labor under the enormous nucleotide hypothesis. This is proposed that DNA was made of a quantity of the four bases. The subunit of this DNA consisted of copy of each basis. Found that DNA of all organisms is one correspondence exists between the purine and pyrimidine bases. This is known as Chagall. Chagov's observation disproved the tetranucleotide hypothesis. The fusion of DNA did not occur in a 1:1 ratio. His results gave insight on and Crick in the development of their model. Please write everything in your paper. Okay. Question about Chagall hypothesis. Do you have a question about Chagall hypothesis? And can I continue? بچه در مورد اصول چارگاف سوالی دارید یا اینکه من میتونم ادامه بدم بعد DNA crystal glass all the time. Watson and Crick were DNA structure. Morris Wilkins, Rosalind Franklin, and were using X-ray crystallography to analyze the structure of the DNA. Tools in the crystal are in an orderly way, so that when a beam of X-ray is aimed at crystal, the beam is scattered in an orderly fashion. This scatter pattern can be on a photograph. The nature of this pattern on the structure of the crystal. The cross in the photograph in figure indicate that the molecule is helix. The dark area at the top and bottom come from the stacked perpendicularly to the main axis of the molecule. The study of the DNA molecule stimulated Watson and Crick understanding of its structure. Therefore, please write this section in your paper. in 1953, proposed the double helix model on two sources of information. Base composition study of Chargaff. In this hypothesis, he indicated that DNA consists of about 50% purines AG, pyrimidines 50% C. T and C are abbreviated of thymine and cytidine, and A and G abbreviated are guanine. B of A was equal amount of T, and the amount of G was equal amount of C. That was Chargaff rules. GC percent content vary from organism to GC percent content varies from organism to organism. For example, cytosine percent, adenosine percent, and thymine percent, and guanosine percent, different organism as Homo sapiens, Drosophila, and Idea is like as you see in the column. Let's write this section because this section is very important for the final exam. Okay. Research of DNA continue. James Francis Crick in 1953 proposed a double helix model based on two sources of information. One, Chargaff hypothesis, and two, X-ray diffraction study by Rosalind Franklin and his colleagues Maureen Wilkins. As you see in this picture, you can see the photograph picture of the DNA crystal that take by Rosalind Franklin. Okay, the Watson-Crick model. With the Watson and Crick constructive models, they possible DNA was one each two helix oil around one other with the sugar phosphate on the outside and the bases on the inside. Again, with the information available, Crick began to construct a molecule model. They found that possible structure of DNA was one in which two helix coil around one another with the sugar and phosphate backbone on the outside and the bases in the sides. Phosphate and sugar outside bases on the inside. خب لطفا این تصویر بکشید داشته باش. Please figure in your paper for better understanding the structure of the DNA. Nucleotides are linked together by the formation of a bond. Nucleotides are linked together, bound by the formation of a between the phosphate and the triple carbon of an adjunct, as you see in this here, you can see binding the figure of the binding of the nucleotides together. Very long string of nucleotides can be polymerized by DSR bonding. Please write this. You can see the mechanism of binding two together. As you see, the hydroxyl group of phosphate section of the nucleotide, hydroxyl group of this section, bonding to hydrogen group of 3' carbon sugar and the final product is H2O. Please draw here in your paper or in your notebook. It is important for final exam. It is very important for final exam. Please write this in your picture. Can I continue? Okay. In this slide, you can see the formation of nuclear chain. As you see, RNA chains are formed through the series of three steps. 1. A base, a sugar is nucleotide. Base attached to a sugar is a nucleoside. A nucleoside with one or more attached is nucleotide. And nucleotides are linked by 5' to 3' phosphodiester bond, 5' to 3' phosphodiester bond between adjacent nucleotide to form a DNA or RNA. Bond formation occurs by condensation involving the removal of H2O and phosphate and pyrophosphates. The structure of nucleoside and trinucleotide with differing numbers of phosphate is shown for DNA. In this picture, you can see the DNA polymerization. Hydrogen binding occur between thymine and adenine and between cytosine and guanine. Between adenine and thymine and guanine and cytosine. Relationship is one of the complementary. There are two hydrogen bonds between adenine and thymine, between cytosine and guanine. Question up to now about the DNA structure and how DNA can binding together and producing a polymerase chain of a DNA. Question? My friend, do you have a question? Okay, if you have not a question, the time of is finished and I will continue your lesson in the next section.